more sophisticated things. So now that leads us on to the. Initially, you started with a vacci bacterial preparation and went on to an OBNC preparation. These greatly stimulate the innate immune system, which have really significant anti-cancer properties, for sure in some cancers, and probably to varying degrees in all cancers. Can you fill out the story of this a little bit for us, Gus? So what started with the vacci and how did that go on to the obiensis, and what are these bacteria and how are they working?
Right. Well, mycobacterium vacci was basically discovered and developed by Graham Rooke, John Stanford, and John Grange. Sadly, the last two are departed, but I worked with them and learned an awful lot. They were trying to develop something better than BCG.
Yeah, which is the tuberculosis.
Yeah, for tuberculosis. Yeah. And they found a bit by accident, and serendipity and accident, that BCG worked much better in areas where there was mycobacterium vacci. So they looked at vacci to see if you could stimulate the immune system.
And this basically comes from cows, presumably?
It comes from cow dung, so
right.
Cynics have accurately called it bullshit.
Yep, fair enough.
And so, but that is, goes all the way back to the so-called interpretation of the dirt theory.
Yes.
You need this dirt to stimulate your immune system when you're young.
Yes.
And as you get older and more sophisticated, you get less stimulation with this. So it was a way of boosting what was already there to give it a boost of the innate immune response. And the big thing is, I've been involved in cancer vaccines for 25 years, and what we discovered is that cancer vaccines, the majority, are useless. I don't really like to admit that, but because that's what I was doing.
But the cancer vaccines that you're talking about there are fairly specific, aren't they?
Exactly.
You're giving a specific antigen, not boosting the immune system as a whole.
Totally 100% correct, because everybody in all the companies went in this. They went for the cancer antigen, the target, MARG, MART, NY-ESO, on and on. And we found when we targeted these that the tumour thought, I'm not having this, and basically, like someone being shot at, ducks, puts a shield over the head, mutates and carries on.
A couple of mutations and you're away.
So this seminal moment is when I was working with Graham Rooke and John Stanford, when they showed that the patients, when they were using for a TB vaccine, that they switched on the suppressed cell-mediated response.
Switched on what, sorry?
Cell-mediated response.
Cell-mediated response,
yes.
Yeah.
So there's a chap called Rossman who basically showed that the immune system is like a big seesaw, and it's a Th1, Th2, he called it. The Th1's a cell-mediated response, which produces things like IL-2, gamma interferon, and that boosts the T cells.
Yeah. These are cytokines that the immune cells release.
Yes.
That boost the cells that actually kill the cancer cells, or kill the virally infected cells, or kill the bacterium.
Yeah. But on the other side of the seesaw are the Th2, and these are the cytokines that produce antibodies and inflammation. And what they found, and then I found the same thing in cancer, was that if you've got HIV, TB or cancer, the cell-mediated response is completely suppressed. It's like someone standing on the brake, and the equilibrium dynamic effect of that is to compensate by boosting antibodies and inflammation. Now, what we found was when we gave these patients MVACI, the equilibrium improved. It was really quite fantastic. Yeah. And I said, this is something that I really want to use in cancer. Yeah. And so I did. Sorry, I'm afraid this is... I just turned it off. I should have thought of that. But
you always forget. You always forget something.
Yeah. So I say I really want to use this in cancer because we've just done a load of really basic work into the immune system, and all our cancer patients are just like the TB patients. The cell-mediated system switched off, and the antibody inflammatory response is overcompensates. And that gives a milieu where these things thrive. Whether it be
it's amazing, isn't it? So cancer can basically turn off the body's natural immune response to itself.
Yes.
Amazing.
Yeah. Yeah. And so I said, can I have this? And can we give it to patients? And these are the days when you didn't have to go through all these regulatory things. I went to the chairman of the ethics. I said, I want to do this. Looked at it. Said, okay. Went to the next ethics committee, signed it off. We did
that's when you were allowed to be a doctor.
This is when we're allowed to be a doctor and do proper clinical research that we can't do now.
So this is the mycobacterium vaccae we're talking about now. This book.
We had amazing responses in some patients. As I've said, these patients were the only patients that had good vitamin D levels, so the two of them. And then we looked at what it did. We found that it boosted these intermediate T cells, the innate T cells. So there were natural killers, gamma delta T cells. And when you read about these things are very, very good at killing tumours and unknown infections before you develop adaptive immunity.
So these natural killer cells, they're big lymphocytes, aren't they?
Yeah.
They would just come up to a cancer cell, basically deposit cytotoxic chemicals on it and just kill the cell.
Yeah.
Simple as that.
And that's why they got called natural killers.
Yeah, great term. They're the killers.
They're the first line of defence. And then thereafter, with all the cytokines and imprinting, etc., you get the adaptive immune response, which interestingly is not nearly as good. This was the insight we had. So until you boost that innate immune response, everything else is not going to work as well. Yeah. Now, going back to all this, we did so much scientific work at how mycobacterium boosts the immune system, and it's all the innate, it's not the other thing. And we found then that, I went to these meetings in the States when they had these checkpoint inhibitors, these drugs which are causing tremendous responses in melanoma, lung, etc., these checkpoint inhibitors. But individually, they never go more than 40%. So I went round this big meeting and I found different groups that said these drugs only work if you've got activated natural killer cells. These drugs only work if you've got activated myeloid-derived dendritic cells.
That's a particular type of white cell that will combat cancer cells.
I said mycobacterium vaccae does both. This explains. So we completed a study and we published it showing if you prime with vaccae, then the response in melanoma patients nearly doubles.
And the downsides to priming with vaccae?
None.
Well, this is a complete no-brainer, isn't it?
Well, this is a complete no-brainer, which is why I'm so devastated that the company that was doing all this has run out of money and gone into administration. Because when I talk about vaccae, we went to Obiense, which is like a twin cousin, but it's actually much easier to manufacture.
So it's still a mycobacterium.
Still a heat kill mycobacterium. This is the thing.
It's a particular process. It's rather analogous to making a particular type of beer. You've got a particular brewery that's got particular techniques, and you get that taste you're looking for.
Yeah, but that is, and so the whole production of this is process, because if you don't heat kill it properly and you don't buffer it and all these things, it won't be active.
It's a sophisticated manufacturing
from a company's point of view, this is a process that they're only not the end product exists around the air, but getting it to a thing where you inject it and you get this great immune response, that's what the company were doing. And they were taking into pancreatic cancer. The whole thing's to me has been a tragedy, because they got the manufacturing sorted out, the FDA had approved all the studies, the trial was up running, just recruiting its first patients.
The trial's actually in process now.
Yeah, so all that development, and this unfortunately is why the company's run out of money, because all this should have taken 6 months or so, but it ended up taking 18 months with all these delays and things with
all the red tape, all the
all the
hoops to
red
jump
tape
through.
and regulations. And
Yeah.
so everything's set up. So I think it'd be an absolute disaster. So I make no bones about it.
Yeah.
It's gone into administration, Imodulan, and we are desperate for someone to come along and rescue it and finish the study and get it approved.
Is there any doubt? Is there any realistic? I mean, the reason you do the study, Gus, of course, is to find out whether it works or not. But all the prognostic indicators in your mind, what's your feeling? Are you pretty confident this is going to be an effective anti-cancer treatment?
I'd like to turn your words around about it, because it's very insightful. The reason you want to do this is to prove it. I know it works. I have used it for 25 years.
Right, there's no doubt about it.
Every single study we have used it in has been positive. And there was a recent a German European group who used it prior to focused radiotherapy CyberKnife. And they have published that this enhances the response and gives them a better response to
radiotherapy.
Yeah. So there's never been a negative issue here. There's never been a serious adverse event of greater 3 than 4, and which in the Pfizer study for the COVID vaccine there were 42,000. I mean, I'm not joking. It's never been one. So we shouldn't have
42,000 versus 0. Yeah, I like the sound of that.
Yeah. So we should not have to be doing this study.
No.
This is this is the regulators absolutely. So it's literally like they want anything useful not to succeed or make it so expensive that eventually, say, Big Pharma gets involved. Because the reason I became so anti the EU was because the European Union Clinical Trial Directive I saw was basically a vehicle for Big Pharma to stop or control all clinical academic trials, all small pharmaceutical trials. Unless they got involved, they were going to die, be dead in the water. And that's exactly what it looks like. So I believe very much that this thing is so obviously works, and all my colleagues have used it. I mean, they love it. They want their patients to be on it. But
I want it because you stop getting colds and flu as well.
Yeah, well, actually, also because of your age, that your innate immune system, in spite of the vitamin D, is not as good as it should be.
It certainly isn't.
Whereas with the mycobacterium boost, it would be back to when you were 50.
If you had it, I would drive down there now and roll my sleeve up and get you to inject it in if it was available. Yeah. It's just not available. So.
I actually believe that the first thing they should do is allow it for cancer studies just to monitor. It's so easy. We did it in a phase 2 study that everybody who was going to have pembrolizumab or nivolumab for melanoma, that if they elected and approved informed consent. Do you remember that? It was totally removed during the COVID vaccine days.
I do. I was injected without informed consent, and I'm livid.
They say, well, I like this. It's got the potential to increase my response from 40% to something higher without any significant side effects. Yes, I have it. Well, that's what the trial showed. And it's so black and white that I believe that any responsible regulator whose goal is to improve the health of the population without exposing them to any serious adverse problems, I believe that they should have released that under a phase 4. You know, we have phase 1 is what is the dose. The irony is that with MVACI and OBNC, it was a bell-shaped curve. So when they did the studies increasing the dose, they found that when you got to a certain amount, the effect diminished. So you reduce the dose, and it really is a case of less is more.
Yeah, you just wanted at the top of that bell-shaped curve, and then
so we know exactly the dose.
And you know that, so you would know how much to put into the syringe to inject into my arm.
To everything. And you know, I've mentioned we've done a randomised study in pancreatic cancer. We've done several in melanoma. We've done it with all sorts of combinations. And I suddenly look back that these weren't trials, but I were able to use it off-label. fantastic stability in stage 4 prostate cancer. We had complete stability in mesothelioma, which is still one of the most difficult, ovarian and colorectal and lung. So this is a generic thing. This is not a one type of tumour or one.
You've described completely different histologies of cancer there, haven't you?
It is. It is across the board, and it makes everything else basically work better, whether it be immunotherapy, chemotherapy, or surgery and radiotherapy. And my surgical colleagues loved me to start the patients on this before they did surgery, because they recovered so much quicker and they didn't metastasize afterwards. And so, which shows that, and then we'd actually already done studies which showed that the bigger the scar, the longer the operation under anaesthetic, the worse the immune suppression was, and the longer it took to recover. So the surgeon I worked with basically said that obvious thing to do is give it before I operate to boost the immune response, and then I'll do less damage. This particular surgeon, who's a really top pancreatic surgeon, Savinda Mudan, who worked at George's and elsewhere, he basically was the first to be absolutely sure that it was really an issue doing the big Whipple's operations on pancreas, because he could do technically superb, but so many.
But that's a sophisticated procedure to take out the cancer in the head of the pancreas.
Fantastic. But it takes hours. It had a big scar in those days. You can do it laparoscopy now, and the immune suppression lasts several weeks. And at that time, several weeks, you totally remove the tumour, but suddenly it appears, secondaries all over the liver. So from a surgeon's point of view, done a technically beautiful operation, but it's a waste of time. Because so the idea of boosting the immune response so you don't get this suppression, so you don't get post-operative secondaries, is a concept that the surgeons embraced long before the medical oncologists. I mean, Donald Morton, who I worked with in the States, who has developed his BCG and tumour cell-based vaccines, he was actually convinced that he could cure people with surgery, providing he could address the immune deficiency. And we're talking like 70s, 80s. He had this insight, along with many others. But the medical oncologists, with their wedded to the chemotherapy, didn't really see much room for immune therapy until the last decade, basically.
So we've got a treatment that's probably effective against preventing and treating a wide range of cancers, a wide range of infections, but our regulators won't let me have it.
Yes.
This is kind of hard to come to terms with. But given that a modulin, which we might call a small pharmaceutical company, is going into administration, and this has got the potential to prevent and treat a wide variety of cancers, can we rest assured that Big Pharma will be so moved with compassion for all the people with cancer that it'll immediately start manufacturing it itself?
No.
No.
No, I'm afraid.
Well, that's a pity.
I am afraid not. And I mean, this substance is so good. I believe it would improve the health of the nation in more ways than one, because of the ability to boost the innate immune system means not that the vaccines are not going to be needed. Now, Big Pharma is wedded to jab. The more vaccines, the merrier. They see it as super profitable. And one of the people I spoke to, and I can't mention names here, obviously, who was very interested in it personally, who worked for a Big Pharma, he said, I think this stuff is absolutely fantastic. I will push it. And then he was incredibly honest. He said, there's an issue for Big Pharma, because it means if we use this, that the costing and everything is limited. It's not an antibody, so we can't sell it for... Some antibodies are sold for £100,000 a course per year per patient. No society can afford that.
No.
With this, it could be super successful as just a few hundred, let alone 100,000 per year. I mean, it really
And work against a much wider range of cancers and infections.
Right. And he actually said to me, he said the problem is that the people who control these companies now are accountants, and they could see this would prevent their ability to use 2 or 3 monoclonals at 100 grand. And eat into the profits because they're only in profit. Now this guy was in the company. He told me that. I mean, it made me think of someone who invented steam-driven engines and bought up by big oil companies and then completely locked away in the basement. It's that type of approach. They don't want anything that might reduce their profits.
The regulators in our country, in the UK, could authorise this. And for say they authorised it for treating melanoma or pancreatic cancer, which would give them clear evidence of efficacy for it. If they licensed it for those things, then that could allow doctors to prescribe it compassionately.
Yes.
For other things. Why don't they do that then?
Well, I don't know why they won't do that, but they can if they wanted to.
It's inexplicable. It's inexplicable.
I was talking about the stages of phase 1, phase 2. We said we've done the phase 1. Phase 2 is does it work? Every single phase 2 study we've done, it clearly is of benefit. Phase 3 is the big randomised study which they want before they register and prove it. But the problem is those large studies cost 20 to 30 million because of the way they did the regulations. Yeah. But there is a thing called phase 4, a phase 4 study. And that means that you license it, but you haven't given it full registration, but people are able to sell it, use it, prescribe it, but you have to monitor it like it was on a study.
Yeah, and you collect longitudinal data in the real world as you go along.
And it's heavily monitored, and if that's fine, then it's approved. Now, I was involved very much with the thalidomide analogues and Celgene, who actually, I've no doubt, would actually have taken this up had they not been bought out by Bristol Myers Squibb. And I fought hard for the analogues, and we screened all these different analogues, and we came up with a drug that's now called lenalidomide. It's one of the most successful drugs in the world for myeloma and lymphoma.
Yeah.
It ticked every box. And I was amazed. They got that, Celgene got that into the market very quickly by using a phase 4 in France. So they were able to sell it, people use it. They said, my God, this stuff works. Everybody did it. In the meantime, they had a phase 3 somewhere else. So they got it. And, that drug is still selling over $10 billion a year. And it was licensed in about 2007. It's just incredible. But they were able to use that then to get it rapidly through, because everybody said that they got the new drug through to approval unprecedentedly quickly at that time. So that's the sort of thing I think that the regulators who said we've gone from regulators to enablers, but that looks like only for COVID vaccines. But they actually ought to remember they said that. June Rain said that. So
enablers of who would come to my mind.
Yes. So they should enable things that, and at the time when Cameron was Prime Minister, that's quite a way back, he actually, I believe for personal things, said that there should be a fast track for drugs that are active in cancer, that are active, non-toxic and cheap. Well,
active, non-toxic and cheap. That describes your bacterial preparation precisely.
And I spoke to someone in the MHRA at the time, said there were 45 applications under this scheme, and ours was the only one that ticked all the boxes. But still, in spite of that, in spite of the applications, no, they wanted another big study that nobody could afford. And this is what drives me mad. There's so many drugs like this out there that would benefit the health of the nation, are non-toxic, cheap, and work at the absolutely root cause of why we get ill, i.e. our collapsing immune response. I mean, the immune response sort of collapses after about 50, and it's amazing we do reasonably well as it collapses, but as it falls down the slope, the incidence of cancer rises exactly like butterfly wings. So you would have thought that if we're going on about the health of the nation, somebody would have thought that this is important. And there are just so many immunologists that would completely concur with that. And we're both friends of Robert Clancy. I mean, he would completely go along with this.
Completely.
So to have a simple agent like this is just a disaster that we can't get it to market. But I believe that one of the reasons I'm here today is that Imodulan ran out of money, but at the same time showed this substance was very active, totally non-toxic, and if it was approved, it could be available widely, cheaply. It would be a fraction of the price of any of the other drugs.
We've got a cheap, safe, effective anti-cancer preparation. This sounds like the Holy Grail.
Yeah, well, the big thing that I was saying before is because with vitamin D in this, you correct the suppressed immune response.
Which caused the cancer in the first place.
Yeah, and then everything else works better.
Yeah.
So the chemo works better, the radiotherapy works better, surgery is more effective and less likely to be followed up by metastatic spread. So it is so obvious to do, but it is incredible that big pharma have looked at this, and the thing that has come across. I mentioned the first guy who says, "Well, it might interfere with our profits." I got it very high up to another big pharma, where the UK group were very high backing, but most big pharma now, the decisions are made in America because they're nearly all American. It's the Americans who run it, and basically it was dismissed as steam-driven, very old tech. We're interested in high-tech medicine. Well, sometimes steam-driven old tech works. Don't overlook it because that's what it does. And I've worked with many high-tech, and when I get really annoyed about the COVID vaccine story, the high tech goes right up there. I've worked on with people on CAR T cells, meant to be the state of the art for liquid oncology. I have been on the scientific board of companies that specialise in messenger RNA vaccines, what have you. None of them are as good as this essential thing, and that is the thing, and they cannot cope with that.
And they're all specific, as opposed to this would work for everything.
Exactly.
Yeah. I mean, how do you? It's a pretty stupid question, really, but basically, you've dedicated your life to medicine, virology, and oncology, and you've basically found, without being too melodramatic, a cure for cancer that's now being buried and suppressed.
Well, I wouldn't say.
How do you cope with the frustration?
The frustration. I wouldn't. I would never say it's a cure.
No. I overstate.
It enables cancer to be managed. Much more successfully. That's what it. And without it, this is the reason why the response rates to a lot of the cancers are 40%, 50. They regard that as marvelous. They're not 80, 90. This is, and I meet so many patients because they say, "Can you help me? We're interested in what you talk about." And they've had ovarian, breast, lung, bowel. They've had horrendous chemotherapy, and it's been
these patients are so ill. It can be barbarous, brutal.
They said, "No, we're not having any more. We just don't want any more."
I've seen that so many times. People that say, "No, I'd rather die than go through that again."
Yeah. So if you actually correct the innate immune response, and there's another agent we found boosts it very much. I think I mentioned it before as a low dose naltrexone because it damps down the inflammation. And I've given lots of patients the two. The result of that is you can give this really tough chemotherapy at half the dose and it's still as good. Well, all the toxicity is in the top 50 to 100% of the dose.
So it becomes way more tolerable if you give the low dose naltrexone.
And I've pleaded and asked patients, said, "Look, listen to me, go on this, and have that chemo at half the dose. And often I have other oncologists involved, and I have to tell them, that they're so programmed. You
Yeah.
have to have the full dose. Well, you only have to have the full dose in tumours that you 100% eliminate. So that's like leukemia and testis. Every other solid tumour, it's not that it's not that linear. And basically, you can control the disease with low-dose chemotherapy, finding the immune response is good, and you keep it going for a long time. And this is one of the reasons we know it is controlled, because people have some big perturbation of the immune system, and that's how I noted the COVID vaccine boosters were causing this, and then it comes back. But
So that could be major stress, that could be infection, that could be vaccination.
All these things. But the bottom line is the proof of the hypothesis is that in so many people, the cancer that's come back is the same one. And in my own patients, I've had up to 18 to 20 years ago. It's not different, it's not new, it's the same one. Yet they've been disease-free for 18, 20 years.
Yeah.
Something happens to perturbate their immune response, and they got the same disease comes back. The only conclusion is that immune system was controlling that cancer perfectly.
Absolutely, because otherwise they could just get another cancer if it was just. Yeah. So giving the low dose naltrexone, does that allow for halving of the chemotherapeutic dose in several cancers or specific ones?
Yes, it does, and we have published this.
It's pretty well across the board then.
Yeah, across the board, and with different chemos in different models. We did all these in standard mouse models, so
right.
What everybody uses, and we could show we got the same effects with the same standard chemotherapy at half the dose if we primed with low dose naltrexone. And I give that to lots of my patients, and I then say, look, I know you really didn't want the chemo, but this can control your cancer, but it will work at half the dose. You won't get the toxicity. And then I will talk to the oncologist who's been programmed. He's got to give it a high dose. I said, no, please don't. And I send the paper, the review. We did a big review on low dose naltrexone that's management cancer, showing how it works on all these different pathways. Yeah. I mean, the thing that really attracted my attention is that we were able and the first to show that it switches off IL-6 production, interleukin-6. And when that was discovered...
Which is a pro-inflammatory cytokine.
Pro-inflammatory, but it was called by the people who discovered it cancer growth factor. So to have an agent that switches off cancer growth factor right from the day one that you add in with your chemo and your immune therapy is fantastic.
Yeah.
And to show how effective it is, there's more than one company produces antibodies to IL-6 that you can use as part of the treatment, usually when it's far too late.
Yeah.
So actually having something that just switches off the fuel supply.
Yeah.
But at the same time, we showed it enhances the sensitivity of cancer cells to the chemotherapy because it reverses the BCL2 Bax pathway. Now, cancer cells hyperexpress BCL2, which is like a shield to cell-killing apoptotic induction. And after LDN, that reverses.
So it keeps the cancer cell alive.
It keeps the cancer cell alive, but it basically removes the shield. So this is another agent which is it was licensed for heroin and morphine addiction.
Yeah, the naltrexone.
In high doses in 1986, it was approved.
So the low dose naltrexone is a drug that you have repurposed.
I'm not the only person. I mean, it was repurposed by Bihari and things. And like most things that I've come to know of, I don't learn anything from my peers or the conferences, nor the literature which they spew out the whole time. I learn it from patients who've done incredibly well, and I'm terribly nosy. And we followed it up. That's how we did all this research.
Absolutely.
And my God, there's a lot of things out there which we have looked at and can't find any scientific justification, so we just put it to a side, etc. So that is how we've discovered how these things are so good.
Yeah.
The naltrexone again it's low dose, non-toxic, cheap. It has minimal side effects. The main side effect is usually if you're taking something with codeine or opiates in it because they clash.
Yeah.
But apart from that, so that's just another example of simple, cheap, efficient things that the establishment wants to ignore.
Yeah.
Whereas I believe the NHS and the regulatory authorities, their duty should be to improve the quality of health care for the population by the cheapest, easiest means possible.
Least toxic.
Exactly, and least toxic, not by giving them injections to reduce their obesity.
And that's without even thinking about the massive financial savings.
Yeah.
So I overstated the case there slightly. I think I've got the idea now that the mycobacterium, vaccae and OB-N can greatly enhance the effectiveness of other treatments. But let's just suppose we were to give this preparation to, let's make something up, everyone at the age of 55, say. What's your thinking about how that would improve health over the next couple of decades?
Well, I would be amazed if it didn't reduce the cancer incidence. And
so we could be talking about a generic or multiple anti-cancer, for want of a better word, vaccine.
Yeah, and also it would protect them from a variety of infectious diseases that we suffer from. One of the things that I noted in my trials of people who've been on IM 101 in 2020 up to early 2021, none of them caught COVID. There was no COVID vaccine. None of them caught COVID.
And presumably would get less cases of sepsis. I mean, in A&E we're dealing with sepsis all the time.
Exactly. That's my point. It is something that if I was running a hospital and people came in and they needed to go to surgery, I would give them this first. If they came in for cancer and they were lined up for standard treatment, I would give this first because it would make it work better.
Yeah.
With this simple thing of your innate immune system collapses, you elevate it back to normal, everything's going to improve. It's so simple.
So if I wanted, well, I do want, but I can't get. If I was able to get this, would I need a shot a year or a shot every couple of years? What's your...
I think as a prophylactic, probably a shot followed by 6 months, then a year, and then probably annually at the most. When we've used it to treat melanoma, we've tended to use the system developed by John Stanford and Graham Rook, where you give it a small injection, you repeat it at 2 weeks, then 4 weeks, and then a month, and then 2 months, but then 3, but then 6 months, and then because obviously you built it up so it needs boosting much less regularly.
Yeah.
Now, what I've learned is the people who respond to it very, very well, and we did all the cytokine testing. If you've got a really good reaction at the site of injection at 1 month, you don't need another one.
Right. So you have an injection and it goes red and painful.
Yeah, well, it's not painful, just slightly tender if you bang it.
You get an inflammatory reaction, and that means your immunity is up to scratch.
And so when it decays and the inflammation goes down, that's the time you need another one.
Yeah.
And it correlated with cytokine. We measure all these expensive cytokine. It correlated exactly with reactions. So you don't need to do that. So that's, millions saved in doing unnecessary tests.
Yeah, and a tiny personal price and a tiny financial price to prevent potentially millions of cases of cancer.
And I've had a patient who had not one but 2 stage 4 cancers. They kept them going for 2 or 3 years with a really bad melanoma, but was completely controllable, but then developed another cancer which was associated with a genetic mutation that almost certainly ran in the family, because this second cancer had occurred at the age of 50 in quite a few people in the family. And we gave this IMM, and we noted that the reaction was very good, but it faded quickly. So we'd have to give it more frequently. And that suggested that the cancer was sort of fighting, turning over, and we could measure that from tumour markers, etc. Whereas people who haven't got that underlying resistance, they've still got a good response. 6 months later, you probably give it to them yearly. Whereas this person, because the big fight with the cancer, we were giving it every 2 to 3 months. But again, this has lasted years, not
people that would have been dead years ago.
Exactly.
But hopefully people like you and me, probably fairly average in cancer risk, every year might be enough.
I agree. I would think. I personally think that it for people who are otherwise reasonably healthy but whose innate immune response is in decline, that if I was giving it prophylactically, I'd be looking at annual.
Right. Yeah. That sounds more than reasonable. And then, well, we're advised to get COVID jabs
because once you've got cancer, that
flu
cancer
jabs
suppress the immune system, which is why it's there.
Say that bit again, sorry.
The cancer has, you've got cancer, it's diagnosed, you got the biopsy, it has suppressed the immune system, which is why it's there. It's fought the suppression. And so boosting that is absolute de rigueur before you can start to attack and get rid of the cancer again. Because if you do it while the immune system's suppressed, you'll reduce it, and as soon as you stop, up it comes again, which is what we know many of these big solid tumours do. The bad breast, the lung, the colorectal, the ovary, bad prostate, all of these can fall into this. Whereas you can boost that innate immune response, then the whole landscape changes for how well you may do in future. And what's happened to a lot of my patients, many of whom were told years ago they only had 18 months to live, is that years and years later they're still here, and we've given them this, and they've run out of the standard options, but they've lived long enough that something else has been developed.
Absolutely.
And this is a really exciting thing about cancer. I've got several patients who lived long enough to be able to take advantage of a new discovery. They respond to that.
So are we open to approaches by investors and entrepreneurs to keep this manufacturing process going?
Yes, I really do hope so, because as opposed to just investing in a company that's got a certain burn rate and a certain value, when a company goes into administration, everybody's lost everything. It's a disaster. All the people invested, shareholders, there's nothing there.
And the expertise and the equipment is all set up just right to manufacture this at the moment.
So people come in and they scavenge. They're like vultures. They go, oh, it owns this equipment. I've been after that. I'll get it for a tenth of the price of buying it brand new. That's what they do. But what I'd like to advertise is that here you can go to the administrators and get the whole caboose. They want to do the whole thing as a...
So someone could buy the job lot.
Yeah, you buy the job lot because you'd get it for a fraction of the price as what you'd have to put in to continue to run it. But you're getting all that last 2-3 years of getting the FDA approvals.
And the trials could carry on.
The trials just take so long with the ethics and getting... all trials have multi-centre. They've all got to do their own ethics.
So the hard bit's done already, really, isn't it?
The hard bit's done. All you have to do is go in, do it, and even the trial has been mainly paid by a company that provides the main drug, which we're trying to show the IMM will actually make the outcome much better if you combine it. So that is why, I mean, I was absolutely devastated when it gone down, because there's no good logic for it. But this is
just a major retrograde step in oncology.
This is pure financial. I mean, usually when companies go down, the product has just gone into man, and it's got, found that it's got horrendous unexpected side effects. You remember like the antibody CD28 at Northwick Park? The volunteers in the Northwick Park.
Oh, I remember that.
Yes, yeah. And they all went, I mean, some of them died, and it was terrible.
Horrendous side effects.
Yeah, they usually go down with, horrendous problems in development.
But here we've got the exact opposite.
It's just totally the opposite. There is no clinical problem. It seems to be effective in anything. There's not been one trial that's questioned its efficacy in 20 years. Not one trial. Every trial's been positive. So why can they not just wrap it up and say, use it, we'll put it on phase 4, which means that rather than just write it out, all oncology drugs are written out in sort of like monthly cycles. But this means every month, instead of saying, are you okay, this, that and the other, you fill a form in, says everything's okay, blah, blah, and you send it back, just like it was on a clinical trial, to the company's central repository. And
so the trial could go ahead if the firm, if Imodulan is saved, but that still leaves us with the problem of the regulators that refuse to regulate it.
Well, I mean, I think if the trial went ahead and it was positive, it would then get regulation.
Right. Is it a well-constructed trial that's likely to give a fair result?
Well, I believe so. I believe it is. The only issue is, I believe they've set the bar unusually high. They want like a 50% bigger outcome than normal, which is really optimistic. The normal is 20 to 30%. That's the only thing I would. When I was taking it over, I'd ask them to reassess, because that's the real world. 50%. We got that in melanoma, but when we got it from 40 to nearly 80, it's double.
But given that there's no downsides, if you said, well, if I give you this injection, John, your chemotherapy is 10% more likely to work, I'd bite your hand off.
Well, put it this way.
Because there's no reason, there's no downside.
Yeah, well, put it this way. I would want anybody that I know, and I mean, I still want them to be able to get this, and the fact I can't give it to them, it really annoys me. And there was a stage there when I could give it, if a trial was running, I could give it named patient basis. So that's
like compassionately.
Yeah, compassionate. That's how happy they were that there was no side effects. And then they came in and said, no, it's for trial patients only. And I don't know why that happened, but it was.
Well, there's precedence for that. The World Health Organization said a particular drug should only be used in the context of clinical trials, even though doctors around the world found it efficacious for treating COVID-19.
Yeah. Well, don't get me on that, because I would disband the WHO if I had the ability
as would I. As hopefully the new administration in the United States will look to doing.
Yes. Yeah. Well, they're certainly going to not fund it.
Yeah.
I think they made that very clear.
That is very encouraging in my view. Professor, as always, fascinating, but drives you mad.
It does. But I just hope there's somebody who listening who might know somebody who's an entrepreneur or a company looking for something like this, which could greatly enhance something else they've got. And it's a bargain basement.
Absolutely.
In which all the risk has been taken. It's been totally de-risked. I mean, there are no side effects, activity in everything. All that manufacturing's been sorted out, which is always an issue when you're dealing with mycobacteria side of things. All that's sorted out. FDA approvals, trial approvals, all sorted. It is a bargain basement. I would say it's the best thing since vitamin D.
And at the end of your life, you can look back and say, you know what? I saved a massive amount of human pain, suffering, and death.
Yeah. Well, so yeah. This to me is just be so important for somebody to come in and do this.
Yep.
Because it's just a tragedy that they ran out of money, but they ran out of money because of the system.
The bureaucratic regulatory system is stacked against anyone who's not got massive big pharma funding behind them.
That's right. And I'm afraid that the big pharma are so powerful, they set their own rules and they bend the rules that exist or just rip them up. Or in the case of the COVID vaccines, they just basically lied about what had not been done, that we find in retrospect.
And a cynic might say that they're sitting chuckling about the fact that this is going down the Swanee.
Yes.
Yep.
But the other thing, we do have, and it is admitted. I mean, we were told you couldn't say. It's been all over the Daily Mail and the Telegraph of the people who had serious side effects from the AstraZeneca and the need for compensation. It's been addressed in the mainstream media. So let us not...
It's starting to trickle in,
let us not mess around. I mean, there were serious side effects which were ignored, and doctors were even struck off for trying to do something about it.
A couple of newspapers. I don't think I've seen it on mainstream national television yet, but
well, it certainly was in Australia, because I was the guy who did it. I did it on the Sky News Australia. That was as big as mainstream you're ever going to get, and they allowed me to do that. They were told that they couldn't, but the people who basically wanted me to do it said, they were told by the government they weren't allowed to do it, but the people behind it basically they knew people personally who had so much damage they could no longer live with it. And we'd had Zuckerberg too say they paid us to censor it, and that people, you can't censor things forever. I mean, that's...
I don't know. I'm not sure about that.
Well,
I'm not sure about that. I think maybe some things you can, unfortunately. Ideas can be buried.
Well,
but that's what we fight against. We're fighting for truth and objectivity.
That's what we're fighting against.
We'll
the whole
keep
point of bringing that up was that the IMM may be the ideal solution for COVID vaccine damage. And
interesting.
And I have a justification for this, because there was a randomized trial published in patients with long COVID with BCG in a placebo.
Yeah.
You see, I can't remember. I cannot give you the.
No, I can't remember the details, but yes, it makes sense.
It was very good. Now I know that IMM would have been far more effective, because BCG is live attenuated, and if you give it more than twice, you get this Th2 inflammatory response, and you lose the benefit.
Yeah.
But just giving it the once, you get that. But with the heat-killed mycobacterium,
that's the difference.
You can keep giving it without ever inducing the inflammatory response. And that it is, if you were trying to work out what you'd need for all these things, it would be the perfect agent. You could spend hundreds of millions developing and decades to do. We've already got it. It's here. That's my message. And it would be fantastic for all this COVID damage thing too.
Yep. So the immunological perturbation, disruption caused by the vaccine, caused by long COVID potentially.
Yeah. This would correct. It is an inflammatory Th2 response.
Yep. Makes perfect. So going back to the vaccines that you and others have developed here, have you got a company that's up and running and saying, we can make these for you now, Gus?
Imodulin is the company. Imodulin is the company making the IMM 101.
And they're ready to go.
There's a chart there. This is one we published in 2016. It is a very simple, multi-center, randomized trial in which the standard treatment at the time for pancreatic cancer was gemcitabine. So all we did was randomize to IMM or a placebo. In fact, not all the places use placebo, because it's obvious if you have a little intradermal injection.
Yeah.
And this is what you're looking at, is survival. I mean, it is unbelievably effective survival, because at the time we used gemcitabine, it was only used for patients who weren't fit enough for the new, more toxic regimens that come in. So that the survival was even more important, because the standard patient we treated was so poorly, and yet we had this fantastic improvement. If you gave IMM and gem, the survival was far superior to gemcitabine alone. I'd just like to add, though, that one other thing that I can never ever tire of saying is that when I was asked why this immunotherapy works so well by a guy in America when we presented it, he said, "Why does none of the other immunotherapies work in pancreatic cancer?" And I said, "Well, this is probably the first trial where we insisted that the vitamin D levels had to get into the normal range." He couldn't believe this. He said, "I've never heard of this, and I'm a very big pancreatic trial cancer person." His name was Daniel von Hoff. And he was he was huffing and puffing about this. But he phoned me 3 months later to say he'd put somebody on a study to check. He'd done 4,000 patients on different trials over 2 to 3 decades. And he came back and he said not a single patient responded to a single chemotherapy agent if they had a low vitamin D level. He was absolutely knocked out. And the really good ones that they put down to the later agents as they came along in the trials, that the responses were purely correlated with the fact these rare patients had high vitamin D levels. So with this, it's one of the things I've always said. We're going to stimulate the innate immune system. You must have a good vitamin D level first.
Which makes perfect sense, because you've got vitamin D receptors in every type of white blood cell. You've got vitamin D genomic expression that depends on vitamin D dependent genomic expression in every type of white blood cell. It seems fairly obvious. Interestingly, though, that the patients with the high levels of vitamin D in the blood, did they actually do better? Was there like a dose response there?
Yes, there was. He confirmed that. He said he couldn't believe it. I mean, he spent 30 years doing this and hadn't realised it was that simple. Well, I must confess that we took a while before we realised that the difference between a responder and a non-responder was the vitamin D. We looked at virtually everything else, and we only cottoned on to vitamin D when we were able to use the hospital's vitamin D testing service that they use for mothers and babies relative to rickets. And as soon as we could do it for adults, we found that it was the same. The only people who responded really well to the vaccines, including the immune modulators like IMM, were the people who had good, healthy vitamin D levels. It also highlighted that the NHS's idea of a normal vitamin D is really too low.
At least in this pathological situation.
In a pathological situation, because vitamin D is too low in the general population, because this is a population that is very white-skinned and exists indoors most of the time, so it doesn't get a chance. And then people who've got darker skins, it is far, far worse. So that they definitely need supplementation.
It's interesting. So for years you'd known that some patients respond to your chemotherapeutic regimes and some don't. And the reason that the non-responders didn't respond is because they were low on vitamin D. It was that simple.
Yeah, it is that simple.
That's so obvious once you know. But so what sort of levels do you like to titrate up to for chemotherapy?
I like to get it over 100 nanomoles per litre.
Yeah.
The NHS normal is 50 nanomoles per litre. So it's far too low.
Yeah, so over 100. But that's still only about sort of 35, 40 nanograms per mil, isn't it? It's not.
Yeah, and the upper limit of normal is usually 200 to 220, so it's nowhere near toxic.
No.
Absolutely nowhere near. And
even then, in your career, have you seen a case of vitamin D-induced hypercalcemia?
It's very difficult to say. My direct question is no. But I have heard of it occurring in children where the mothers are given ludicrous high doses because the kidneys can't take it. You've got to be very careful giving vitamin D supplementation in renal failure.
Right.
And certain people with rare phosphate genetic pathway abnormalities. Apart from that, you normally just pee it out. If any excess, you have a thermostat, and once the vitamin D goes up, I mean, there's some people you can give grams to nearly, and they'll never go above 120.
Interesting. So even though it's a fat soluble vitamin, because traditionally I would teach that it's water soluble vitamins that are urine excreted. But here we have a fat soluble vitamin that's actually urine excreted. You can test the urine and find high levels of vitamin D in the urine if you give large amounts. It's just it just seems complete no-brainer not to do that. Is this done now in cancer centres around the country? Is vitamin D replete?
It's slowly, but I just cannot believe the number of people I get referred for second opinion. Will you please see this person? They've been on all the right treatment, they're not responding. Have you got any sort of magic, this, that and the other? And I find they've never had their vitamin D measured from, big cancer centres, which I won't name. And I measure the vitamin D and it's really low, and so that's the first thing I do, boost it up. And I thought that should not be my job.
No,
it should.
You should get to a professor to do that. The junior should do that.
Well, exactly. Anyone comes near me should have a very good vitamin D level. It's not my job to point out you've just had a year of treatment. It's a total waste of time because you've got a low vitamin D level. It's unbelievable.
And the human suffering and the financial cost involved in that. It's just unbelievable.
But even hospitals with very good reputations are not getting vitamin D measured in a lot of their patients.
Yeah, incredible.
In fact, the people who do push for it are actually the dietitians.
Yes.
Not the doctors, not the oncologists.
Yeah.
The oncologists will say, well, there's no good evidence. Of course there's good evidence. They just never ever bother to do the research on it.
So two very natural things: the IMN, which is the mycobacterium attenuated bacterial preparation; the vitamin D, perfectly natural, and getting those two things right has got this massive benefit. Maybe just look at so that's pancreatic cancer. What is this one?
That one was for lung, but it came from an earlier study which was negative. I have to point this out, but this to me shows the advantage of looking at sub-selection, different components of a trial. It is poo-pooed by statisticians. They say you shouldn't do it for lots of reasons, but this showed that if you just looked in this group of lung cancer patients who overall had no real benefit from having IMM added into their chemotherapy. But if you actually looked at those patients who'd had 4 courses of the immune stimulation, which meant that they had survived 3 months or more, and you then looked at the subtype was either squamous or adeno. We found for the adeno, it actually had a benefit which had been drowned out by the bigger study. And the adeno for me was important because most pancreatic cancers are adeno, which is what pushed us to use the agent in pancreatic cancer.
Right.
And then I've done it in prostate cancer too. It's fantastic in prostate cancer because most prostate cancers are adeno. The vast majority. But we've done a great series of anecdotes but could never get any interest in doing a proper study in prostate cancer patients. A cynic would say presumably because it's so uncommon, but there must be other reasons.
Yeah, you never see a case of prostate cancer, do you? It's let's look at one more maybe. What's this one?
Well, that was the original. That was
the melanoma.
We did in melanoma, and these were patients. Some of them being on trials, randomised with other treatments, and the vast majority were not randomised when we saw a very strong signal with it. So the green line shows our 5-year survival is to 60 there halfway, and it goes out to 10 years, as you can see.
Yeah, it's impressive.
Now, at the time we commenced this, that survival was the yellow line.
Wow.
Which is, it's a massive difference. In fact, the difference is nearly as big as for ipilimumab, which is the first immunotherapy to be approved and which started to revolutionize it. And that's another interesting thing that we showed. If you gave the patients this IMM 101 or VACI first, and then you they relapsed, and you had to give them one of the new approved immunotherapy agents, which had a 40% response rate. And we published this recently, my colleagues published it. We showed if they were primed with the innate immune boost, and then they had the checkpoint inhibitor, Pembro, Keytruda, Nivo, Optivo, the various, they're all roughly the same. The response rate was nearly double with no added toxicity.
Because you're upgrading the immune system ready.
Yeah. So that is a fantastic outcome. Again, we cannot get the big pharma to be interested in this, even though they're aware of it. It's rather like they just don't want to know.
Interesting, the possibilities of the IMM, the mycobacterium vaccine, combined with vitamin D and ivermectin. I wonder how many cancers that would treat.
Well, it's a study I would love to do. You would call it a basket or an umbrella type of trial. It's just all comers. You just add it in, and then you would just monitor: are we getting better responses? That's what I would love to do, because you have you start from a position, unlike COVID vaccines, that they're safe.
Yes.
Years down the line, there are no SAEs above 2 for any of the studies, and they're over. I calculated there's about 4,000 patients have had Vacci or Imodulin, because it started out in the big TB studies, and so there's no significant side effects. Just at the site where you give the intradermal injection, you might get a big reaction there, although that's good. It means the immune response is working to it. Some people worried it gets infected, and we've swabbed hundreds of them. They're never infected. What appears the infection is a very strong macrophage, which has very strong anti-tumor activity response. So
the inflammatory response just shows that it's working.
And we showed that it correlates with cytokine changes that you measure from blood in your arm, which are very expensive to do. But we showed they correlate beautiful with reaction on the arm. So once the reaction on the arm is really settled down, if they've got a cancer, you give them another one.
Yeah, the cytokines show you're getting a systemic benefit as well. Yeah.
So it's incredibly practical and pragmatic.
The other thing that a lot of people feel strongly about, and I do, if people have got, say, stage 4 cancer, then there is no reason not to try these things. So anyone with stage 4 cancer, prostatic cancer, for example, we think of the tragic news of Sir Chris Hoy over the past few days. 48 with bony metastases from stage 4 prostatic cancer. I mean, anyone in that situation who wants to try ivermectin and vitamin D and IMM should be allowed to.
Yeah, I agree.
And then these patients would opt into this, and then you would develop a cohort in no time at all, because patients would be opting in. Compare those to the patients that didn't opt in, and I strongly suspect you'd see a very profound difference in mortality within months.
Well, I mean, I've got prostate patients, not ivermectin, but the IMM aspect, who have been completely stable since starting on it. I mean, just no progression, whereas before they were having progression. So just that agent, and vitamin D, of course. But and then the other one I've been using, which is like the ivermectin, because I can get it, is the low dose naltrexone, which I mentioned before, because that damps down the chronic inflammation that is associated with cancer spreading. So I would actually include that as well, and then with what we've learned with the ivermectin, that none of them, I don't think, would interfere with each other.
Interesting to hear you say that they wouldn't interfere with each other. And as we find that these drugs are efficacious, potentially we could start titrating down the more conventional chemotherapeutic agents, the more specific ones. We don't know that. That's pure speculation. But
we don't know. But what I can tell you is that in my career, I've lost count of the number of people who saying, I'm not going to have chemo, because they knew people who had terrible
Horrible.
responses with it.
Yeah.
Or I've persuaded a lot of people to have chemo on the grounds the chemo I use, like for melanoma, it's not that toxic if you play it right. And so, you're thinking of people who've had really high dose aggressive treatment for breast or lung or something. It's not like that. But I've met so many people who've just said, no way, I'm not having it, I'll try anything else. And more recently, you might remember being brought to your attention, but Ellie Macpherson was in the Daily Mail having said that she'd got breast cancer, and she had looked and done a lot of research and wasn't going to have any chemotherapy herself. So even some high profile, very intelligent people are saying, no, I'm not prepared to go down that route.
Yeah, they'd rather die than suffer that amount of. Yeah, absolutely. But again, to these people, ivermectin should be available over the counter, as far as I can see, and they're simply free to try it, collect the data, then would know. It's just
yes, as I remember my wife saying she could get it over the counter for scabies for the children, and that's mysteriously disappeared.
Ivermectin does seem to be in really short supply, despite the fact that they can produce it by the ton in drug factories. It does seem to be getting very expensive and hard to find.
It's ridiculously expensive, and I think this is pharmaceutical manipulation. I'm prepared to voice my speculation, because there's no other logical reason for why something that can be produced in the tons, sold for pennies, distributed to millions of people a day, should suddenly become difficult and extremely expensive in the West.
Yep. My last ones I imported were from over the counter from India, so it's still we know it's still available, still produced. So it just seems to have difficulty diffusing around the world these days. Do you want to mention this about the vitamin D positive results?
I mentioned about it boosting the checkpoint inhibitors, and I put it the third line, which says IMM enhances MDC. That means myeloid-derived dendritic cells and natural killer and gamma delta T cell. I was at a presentation.
These are all immune cells.
Immune cells.
Protective immune cells.
Where they different people have done this work, and they presented when I was at an immunotherapy conference just before the lockdown occurred in America, and I was staggered. People showed that if you didn't have activated myeloid dendritic cells, the checkpoint inhibitors wouldn't work. And they said, this is what you've got to do to make them work. And they go to another demonstration another day, and another group would say, here's the secret. If you don't activate natural killer cells, the checkpoint inhibitors won't work.
And they can only be activated with the vitamin D.
Here we have one agent that did both of this in-depth research. And we've shown in a small study that it doubles the response rate. So almost certainly through these mechanisms that others have elucidated so comprehensively.
Yeah. So there's, in your view, there's no oncological treatment that would be adversely affected by making a patient vitamin D replete prior to the commencement of treatment?
No, none.
There's no contraindication here. All patients should be vitamin D replete. Full stop. That's the end of it.
Period.
Yeah, absolutely. Yeah. What's this?
I think we can jump up. Well, actually, this is very interesting.
No, go for it. Yeah.
This was a paper published by in Nature Medicine by Gentles and the group, which just pure looking through the literature, what cells control cancer. Now, the entire industry of immunotherapy thinks it's CD8 and CD4, but this showed quite categorically.
So CD8s are the T helper cells, and
the gamma delta
killer
T cells at the top with the arrow there. The gamma delta T cells. Now, my group has spent the last over a decade ago, showing that gamma delta T cells are the ones that you really want in cancer, and they've shown that beautifully. Daniel Fowler and others in a series of papers, and here we have something that confirms it just from an observation status. They didn't go out to show that. They just said which ones are the most effective, and they found it was the gamma delta, and of course the IMM boosts that gamma delta cell beautifully, where most other immunotherapies don't.
So to put it simply, these gamma delta cells are a subset of T cells.
Yes.
their activity is boosted by giving a few dead bugs intradermally.
Yes, the gamma delta cells are basically they are a cell midway between the very primitive T cell that has no recognition of anything
Right.
particular,
Yeah.
and the adapted T cell,
Right.
which when mature will
Right.
see a specific antigen through its
Right.
cytotoxic T
Right,
cell
right.
lymphocytes will kill it. It's halfway through there, and it would appear that this is far more important than the highly evolved, sophisticated pathways which we've all been trying to manipulate.
Yes.
So once again, it's a simplicity which is so attractive and exciting because it's very practical to do so.
So we have these fairly crude cells. I'd call them natural killer cells. Is that right?
Yeah.
Fairly
It's just,
big lymphocytes.
just slightly more evolved than the natural
Yeah.
killer cells. Yes, they can
But obviously, we've got a spectrum of these in the body,
Yeah.
precisely for this reason.
exactly.
It's amazing, this spectrum of cells from a rather crude thug that goes around beating up any cell it finds to one with a very precise antigenic target. And we have a spectrum of these. And because the mycobacterium IMM is polyclonal, it's got the potential to stimulate many subgroups of cells.
Without a doubt. I mean, there's so many different types of antigen in the mycobacterium. There's sort of lipids and gangliosides and all these various different presentations. And I think that's why it is so effective, because if you set out to make a product without using this as the stepping stone, you would never think to add all those things in together. You'd only ever do one or two at a time.
And that's the big mistake. Well, it's not the mistake, it's deliberate. The pharmaceutical industry is targeting one very precise thing because that's patentable. Whereas patenting a bacteria would probably be slightly more tricky for them.
Well, like the protections in the manufacturing process, that's really where all the skill set is.
Yeah.
Because it's very easy to produce a load and it's not effective. So that's where the skill set is in preparing these.
But your mates could. The skill's ready to go. If they press the button, they could produce tons of this
stuff.
stuff.
The company, Imaginem, have got the patents to protect this.
So Imodulin could make enough to vaccinate the whole country within a few years if the government said go.
Yeah, exactly. Yes. The beauty of this is that you don't need to resort to gene therapy, as I call messenger RNA. You don't need to resort to this to produce millions of doses quickly. These things grow so fast that you can produce millions of doses quickly. You don't need
just like making beer, isn't it? You just chuck a load in and culture them. Yeah.
Yes, that is the best. It's like yeast.
Yeah.
Just like yeast.
Yeah, I mean, they'll presumably divide every 40 minutes or something, and get infinite amounts. Now, so just going back to the pathology we were talking about before, we have these things in the body called tumour suppressor genes. And that's what they do. They're going to inhibit cell division and inhibit the formation of tumours and cancers. So having healthy, active tumour suppressor genes is going to greatly reduce my chances of getting cancer. So I want my tumour suppressor genes to be as active as possible. And the idea is that if the spike protein can inhibit the activity of the tumour suppressor genes, then what is left to suppress the tumour? And you mentioned the specific examples there, MHS3, P53, and I think BRCA2 is probably another one, that BRCA, breast cancer.
I saw it mentioned in a review. I hadn't seen the primary data on this, but that's a suppressor gene. That's the one associated with breast and ovarian. So people who have BRCA mutations are much more likely to get breast and ovarian cancer before they're 50 than those that haven't, which really upholds the model of what I was saying. Interfere with these things and your cancer pops out a lot earlier. And that's what I'm worried about.
So these suppressor genes are exactly what we want to be healthy, but they are potentially being inhibited. And the other thing the paper pointed out was that the modified mRNA vaccine contains N1-methyl pseudouridine. Now, this is one of the bases in RNA, and it's not the natural uridine, it's pseudouridine because it lasts for much longer. And that means the spike protein could be produced for a much longer period of time. again, potentially going on to inhibit more activity of the tumour suppressor genes. It really is quite a frightening potential scenario.
It certainly is. Now, again, when I pointed all this out, I've, people say, shut up, you're just a cancer doctor, you don't know what you're talking about. And that really gets me wild, as you know, from all my HIV experience. But one of my other things, I've been on many scientific advisory boards for grants, authorities and companies. And I was on the scientific advisory board for a company that, after its name, it says the messenger RNA vaccine company. I was on that board for 5 years. And I knew there was a big problem because messenger RNA gets dissolved and killed and mopped up in 20 seconds if it's in the body. So you can't possibly use it for a vaccine until you stabilise it. And the very fact, if you stabilise it, the big problem is it's stabilised. So that is the fundamental flaw with RNA vaccines.
And carries on for an indeterminate period of time.
Banned. I mean, no vote. They should be completely banned. If they're going to be used for anything, it has to be what they are, gene therapies. And you might use those in advanced cancer and things like that, but you should never use them in general population as vaccines because they're not.
Yeah, because in advanced cancer, someone's life is already at risk, so we can carry out more risky treatments, potentially. It's all about risk-benefit analysis. The paper also talks about suppression of cancer immunosurveillance. What is cancer immunosurveillance and how could it be suppressed?
Well, I think that's what I was mentioning about the T cells. Yeah. It's just suppressing the T cells, and that's what they do. And if you do things that suppress the T cells, up you pop with cancers. And the ones you're most likely have got along that progression line, sitting at 5 out of 6, they're the first ones to pop up. And HIV showed us that. It was just an incredible model. So you suppress the T cell surveillance, up pop the lymphomas. Because the lymphomas associated with EBV, Epstein-Barr virus. Everybody is infected with Epstein-Barr virus. Only about 1%, far, far less than 1% ever get a cancer with it. But what that did is remove the T-cell suppression, which is T-cell surveillance, which was keeping it in check. And the other one that popped up was Kaposi's sarcoma. I mean, that really popped up massively in HIV. And there wasn't a virus associated with it. But they thought there must be, and they found it. And that's called human herpes virus 8. And left to its own devices, it does nothing. It'll induce change, some nasty changes in the skin, etc., but it won't kill you. But you lose your T-cell ability to suppress that virus, and it comes out with great angiogenic Kaposi's sarcoma that has actually throttled people. I mean, it's so out of control, it'll actually throttle them so they can't breathe. I mean, it's just unbelievably horrible. And that's never been seen until the HIV came along. But we know the principle because you see these diseases and skin cancer, what have you, in patients who are heavily suppressed and long-term suppressed for transplants, etc. And then you have to modify that, etc. So that's what's meant by the immune surveillance. It's purely the T-cell and the innate immune system that are doing it. And what it tells you is what I've been arguing from the beginning of my oncology career is the most important thing in controlling cancer is the immune system. And I was always told, no, it's not. No, it's not. It's you have to kill them with toxins. I mean, everything was chemotherapy or radiotherapy, and the result was all the same as total destruction. It didn't entertain any role for the immune system whatsoever.
I mean, I would imagine at our kind of age, I probably get cancer most days. Do I? Is that reasonable? It's just the immune system keeps it down or eradicates it.
Most people speculate. I mean, people go into this. Some mathematicians will even reduce it to the number of cells that might mutate arise in your body every day, and the T cell system controls them. And I mentioned before that I think that it is the innate immune system that controls them, as opposed to the highly adapted one which vaccines are meant to induce. And the thing that concerns me, and if I was Chief Medical Officer, I'd want to do something about it, is that highly important innate immune response falls off when you're about 55. It really starts giving up. And by the time you're 70, 75, it's in your boots. And if you transform a figure for the cancer instance of that age range, it is on the floor about 50, 55, and it goes up and up and up and up to your 70, 80. It's not necessarily causal, but the association is so tight.
And it's a perfect inverse correlation, isn't it, as the cancers go up?
Correlation. So
in exactly the same way the T cells go down.
In fact, when you draw it, it looks like a butterfly wings.
Yeah.
Yeah. So obviously, to help control cancer, the best thing to do is try and boost that innate immune system as it declines. It's involved in everything, with the susceptibility to infections, bizarrely cardiac disease, but definitely cancer. And doing something about it and addressing that, I think, is one of the most important things that a chief medical officer could do, but they don't seem to be interested. We do know, and you've had other guests here who've told you the most important thing everybody can do, and the government should do it, as everything they orchestrate and conduct they seem to make a mess of. Vitamin D supplementation to good high levels is the most important thing that can be done to stop the innate immune system doing. They don't do anything about that. And when they do come out with some inane comment, they say, well, all you need is 400 international units for
nothing.
Your bones. No, we're not talking about bones. We're talking about the immune system and the surveillance of these other diseases. That does really annoy me. Then the other things, it can be stimulated with mycobacterial things of various descriptions, and I focused on a heat-killed mycobacterium, which I've discussed here before. Now that is so easy to make and do that I would again look at that. They put so many hurdles, and I noticed there was something in the paper only this weekend that it's still impossible to get for small companies to get sensible things through. For cancer, the hurdles are so high and so prolonged, costs a fortune. This is why these companies go and collapse. Yet you come along with something like an unproven, untested, with a heritage that every time it's been tried in any other form, it's been a disaster, and all the things they go, we want to make sure it's safe. And we must have more. This is what they told me. They told me about that. It needs more work in animal models.
Yeah.
This is after it's been in patients, hundreds of them without a single side effect. Yet they didn't seem remotely interested about the side effects or safety of the vaccines that they poured out. It's completely discombobulated. I personally think the MHRA is unfit for purpose and should be completely shut down, as I do with the Department of Health and the NHS that run it or meant to regulate that too, because it's been an absolute disaster. It's a disaster for what we've had to go through with the vaccines, but it's a disaster is that they seem to make it so difficult for anything that is effective, safe and cheap.
Yeah,
they're not interested in. You've got
you've used this. It's basically a BCG type preparation, isn't it, with bacteria, and you've kept patients alive with this for decades. It boosts T cells, and yet, if it was available, I would get the train or drive down there to you now and be injected with it, because I'd love to boost my T cells. But the Medicines and Healthcare Products Regulatory Agency won't license it for compassionate use. It just seems outrageous.
I think
I'm being deprived of. You could boost my T cells tomorrow, and yet you're not allowed to. That's just outrageous.
They won't let the company release it for compassionate use or off-label use, when that has been a pattern which used to exist, but it's under the guise of safety. When there are literally thousands of people have been given this and similar things in TB trials with no side effects whatsoever. Yet they bang on about side effects and safety issues and stuff. I mean, it is a farce. It is a total farce.
And if you were allowed to boost my T cells, I'm less likely to get a whole range of cancers, probably get less infections.
Yes.
And it's safe. And in fact, to call this a vaccine is misleading, because this is giving some borate buffered, from what I remember, heat attenuated bacteria, and to use the same title for that as these wretched mRNA genetic concoctions, to me is quite an outrageous conflation.
We actually call these the category they fit in as immune modulators.
Yes.
As opposed to vaccines, because vaccines now got a really dirty name.
Yeah.
And the COVID vaccines are COVID. What BioNTech says on their first sheets, they go COVID vaccine and gene therapy. They actually have gene therapy written on there. I was staggered. It's that blatant in your face.
Yeah. Yeah. I've just got really one more question in mind. From your experience, what blood levels of vitamin D are desirable in cancer management and cancer prevention?
Over 100 nanomoles per litre, which is twice the level that the NHS say is normal. So
that's about 40 nanograms per mil, isn't it? Roughly that in American. Over 100. That is really useful, actually. So GPs around the country should be testing this and titrating everyone up to that level, I would have thought.
I first picked this up when I was doing very early cancer vaccines, and then we had the immune modulators. The vaccine implies it induces a particular immune response against a certain particular antigen. That's a vaccine. Now, the IMM mycobacteria, they don't do that. They induce an activity of the innate immune system that then allows a T cell to see something that it hitherto hadn't seen. It's like putting glasses on it, and so you then see it, and you get a knock-on effect, like domino effect, by doing this. The problem with cancer vaccines, as I pointed out, and I was a big fan of it, and I very quick to want to know what works and what doesn't work, is cancer vaccines only work for a few months. The simplicity is that when you get resistant to chemotherapy, the tumour gets resistant and goes on another path. The same thing happens if you immunise it against a certain antigen. Of course, we can control them in mouse models, and you'll see improvements in the humans, but it only lasts 3 months or so, 6 months at the most. So what's the point of that? You want something better. You might be able to use it for a focused, targeted beating up of a residual cell. But if you don't do it within a certain time, the tumour is just going to mutate and escape. So that is why we've got to be very careful with the word vaccine, because in both cases it's very misleading.
Yeah, but if we use one of your immune modulators, that's not specific. That's going to boost the body's own T cell responses, and the T cells are already pretty smart. There's just not enough of them. You know, the T cells are part of creation. They're brilliant. So whether you believe they're created or evolved doesn't matter. They're amazing physiological cells that can do all sorts of things. We need to boost them a bit.
Yeah, without them you die. Yeah. And as you get older, they give up before you do. So it's a logical thing to give them a bit of help.
Yeah. Well, as soon as this immune modulator to boost my T cells is available, I'll be down there like a shot. And I would appreciate you giving me one.
We need people to lobby the MHRA and whoever is meant to control them.
We really do.
We really do, because they're not doing their job on anything at all.
It's outrageous. And it's also very consistent with Professor Clancy. I know you're familiar with his work in Australia, giving immune attenuated bacteria that then goes to the pest factors and goes off to promote mucosal immunity.
Yeah. It's the same principle, just different ways of doing it.
Yeah. So I think practically, people need to. We don't want to frighten people, but we do want people to get treatment as early as possible if cancers do develop. So I think really it's a good idea for people to be aware of potential cancer signs in themselves and their loved ones, and really go to the doctor as early as possible if you have anything that you are uncomfortable with, and get early treatment, because that's going to massively influence the outcome of the disease.
Indeed.
Yeah. Professor, as always, fascinating, mind-blowing, a little perturbing, but the thing I've really enjoyed and benefited from on this talk was the complete integration and harmony between your medical experience and the science. They just mesh together beautifully, which of course to me means that both are much more likely to be correct, because they have complete consistency. So it's a Sunday afternoon. You've given me a lot of your time, and we're very grateful, and thank you very much.
Okay, thank you. Bye.
Thank you.